Students, our today's topic is on apomixis, again on plant embryology. First, we define apomixis. Apomixis derived from two Greek words apo and mixis. Apomixis is a type of a reproduction in which sexual organs of a related structures take part but seeds are formed without union of gametes. This process known as apomixis. The first discovery of this phenomena is credited to Leeuwenhoek as early in citrus seeds. The genotype of the embryo and resulting plant will be the same as the seed parent. This is also known as clonal seed production. Modes of reproduction. In this flow chart, we describe two types of modes of reproduction. One is sexual and then asexual. Asexual type of reproduction further classify into vegetative, and one method is apomixis. Apomixis is further classified into two types, gametophytic and sporophytic apomixis. Development of embryo without sexual fusion. Genotypes of developed plants are identical to the parent plant. This is the most important feature of apomixis described by Leeuwenhoek in citrus seed. Types of apomixis, two types. One is gametophytic, another is sporophytic. First, we study gametophytic. Unreduced cells give rise to a megagametophyte. Three types: diplospory, apospory, and androgenesis. Sporophytic type of apomixis, unreduced cells give rise directly to an embryo. This is also further types: adventitious, parthenogenesis, and apogamy. Gametophytic apomixis. If the unfertilized cells give rise to a megagametophyte. First one type is diplospory. Megaspore mother cell MMC cell give rise to a megagametophyte. Apospory. The embryo sac originates from the any diploid cell except megaspore mother cell either directly by mitosis and or after interrupted meiosis. Androgenesis, third one type. The embryo sac originates from generative nucleus of pollen tube cells, the most common mechanism of apomixis in higher plants, characterized by the presence of multiple embryo sacs. Sporophytic apomixis. If the unfertilized cells give rise directly to an embryo, this is known as sporophytic apomixis. Haploid parthenogenesis. Embryo develop from egg cells. Haploid apogamy. Embryo develop from synergids or antipodal cells. Adventitious embryony. Embryo directly develop from nucellus or integuments. No production of embryo sac. Mango citrus. The diagram shows the process of double fertilization, sporophytic generation, and gametophytic generation. In this diagram, various pathways followed by different cells of embryo sac leading to different forms of apomixis. Types of apomixis based on occurrence. Another type of classification of apomixis. According to this classification, first one is non-current, recurrent. Embryo develops from any cells of embryo sac. known as non-recurrent apomixis. Haploid parthenogenesis, embryo develop from egg cell. Haploid epigamy, embryo develop from synergids or antipodal cells. Recurrent apomixis, embryo sac, embryo develop from without meiosis from diploid cell. Embryo develop from MMC, apospory, embryos develop from any vegetative cell of ovule, then embryo develop from diploid egg cell. Adventitious embryony, also known as sporophytic budding, embryo directly develop from nucellus or integuments, mango and citrus. The ideal apomictic system, all the progeny of plants should be apomictic so that progeny have the same genotype as the maternal plant. The apomictic genotype should preferably be fully male, fertile and self-incompatible and reproduce via pseudogamy. In case of diplospory, chromosomes should not pair or recombine during first mutate division. Apomixis should be dominant over sexual reproduction. Expression of apomixis should be little affected by the environment. Development of apomictic lines for various purposes. Apomictic lines can be developed by the following three different approaches. Gene transfer from wild species. Genes controlling apomixis can be transferred into a crop species from a related wild species. From Trypsicum dactyloides into maize, from Panicetum orientalis into permillet. Induced mutations. This approach aims at developing apomictic form in normally sexually reproducing species by utilizing induced or even spontaneous mutations. These efforts have focused primarily on sorghum, where two mutant lines showing facultative apospore. Isolation of apomictic recombinants from interspecies crosses. Sometimes apomictic recombinants can be recovered from segregating generation of crosses between two sexually reproducing species. For example, Estivum and Avena sativa, Odium vulgare and Triticum estivatum. Importance of apomixis. Role of apomixis in plant breeding. Rapid production of pure lines. Apomixis is an effective means for rapid production of pure line plants. Maintenance of superior genotypes. Apomixis is useful in maintaining the characteristic of mother plant from generation to generation. Conservation of heterosis. In some cases, hybrid vigor may be conserved for many generations by using recurrent apomixis method. Advantage of apomixis in planting breeding process. Rapid multiplication of genetically uniform individual can be achieved without risk of segregation. Heterosis or hybrid vigor can permanently be fixed in crop plants. Efficient exploitation of maternal effect in plants, if present, is possible from generation to generation. They may be shifted. Homozygous inbred lines, as in corn, can be rapidly developed as the produced sectors of diploid tissues and occasional fertile gametes and seeds. Advantage of apomixis. Oligate apomixis permits fixation of heterosis in the hybrids. Therefore, farmers can resow the seeds produced by apomictic hybrid generation after generation. The new hybrid varieties could be multiplied from few hybrid seeds in the same manner as is the pure lines. This greatly simplifies hybrid seed production. Even such parents that flowers at different times may be crossed in a greenhouse. to obtain few hybrid seeds which can be used to establish the new hybrid variety. The nuclear seed of hybrid varieties can be conveniently maintained as hybrid varieties. Problems associated in utilization of apomixis. Apomixis is a very complicated phenomenon. The process very complex. Integrity. Estimation of the level of facultative apomixis is tedious and time consuming process. In case of facultative apomix, the proportion of sexual progeny is affected by environmental factors like day length and temperature. In the absence of morphological markers linked with apomictic development, maintenance of apomictic stock becomes very difficult. The genetic basis of apomixis is not clear in most cases. These are the some facts about apomixis. Thanks, students.